When we think of crystals, we often picture gemstones or minerals, like the ones found in this stunning geode. But crystals are not limited to minerals or diamonds. They are all around us, even in our everyday items. For instance, your morning cup of coffee contains crystalline caffeine and sugar. Your ceramic mug and metallic spoon are made up of densely packed crystals. Some of you probably like iced coffee, and ice is the crystalline form of water. And I know we all used our phones during breakfast, and the very foundation of modern technology relies on silicon crystals. But it's not just inanimate objects. We make crystals inside our bodies, especially after a few more cups of coffee. So this brings me to the question, what are crystals? By definition, a crystal is a solid material whose constituents are arranged in a highly ordered microscopic structure extending in all directions. Simply put, a crystal is a solid with symmetrically arranged atoms. But have you ever noticed that some crystals seem to grow into specific shapes again and again? Take this pointed quartz, for example, or this pile of salt cubes. As it turns out, the atomic structure is a defining feature of the crystal, and crystals grow into specific shapes which reflect this symmetry. So salt, which we all have in our kitchens, has a cubic crystal structure, while quartz has a hexagonal one, and these are reflected in their final shapes. However, if a crystal has a characteristic shape, how is it possible that no two snowflakes are alike? This is because environmental conditions during crystal growth greatly affect the crystal shape. Temperature, duration, or even other materials that are in the vicinity can have a great impact. But this is important because the different faces of the crystal can have different properties. You can see here that some faces look shiny while the rest are transparent. Therefore, by controlling the crystal shape, we can control their properties, like reflectivity. A great example of crystal shape affecting optical properties can be found in nature. Apparently, animals also make crystals, some of which they use for their coloration or to enhance their vision. And though it isn't obvious, all of these animals here, from plankton, spiders, reptiles, and fish, all use the same highly reflective material to make their crystals. They are all made of a molecule called guanine. Let's take spiders as an example. Okay? Here you see the white widow spider, the silver desert spider, and the green-eyed wolf spider. Each of them uses the same material to make their crystals, but because they use them for different functions, they shape them differently and also assemble them differently within their cells. We try to make crystals as perfectly as nature does, but even microscopic plankton do it better than us. Discovering the tricks that organisms use to create crystals is important because molecular crystals like guanine play essential roles in our science, technology, pharmaceutical, and food industries, where it is paramount that we control their properties. And learning from nature could help us make these industries more environmentally friendly and less polluting. So now let's get to know the hero of my PhD thus far, the scallop. Here you see this adorable scallop swimming away from a diver. You might ask yourselves, how does the scallop even know the diver is there? Well, it isn't very well known, but adult scallops have about 200 bright blue eyes lining the soft tissue between their shells. Each eye is about 1 millimeter in diameter and contains a curved mirror made of guanine crystals at the back. When light passes through the eye, it is reflected by the inner mirror back towards the photoreceptors, creating an image. This is a lot like a reflecting telescope. And this enables the scalp to see and respond to its environment. In order to study the mirror, we use scanning electron microscopy, which enables us to see tiny things 1,000 times smaller than the diameter of a hair. And in order for biological samples to remain intact during imaging, we use the microscope under cryogenic conditions at minus 120 degrees. This way, we can see that the mirror is made up of thousands of ultra-small square guanine crystals arranged into these tiled layers, and they span the entire diameter of the scallop's eye. But in order for us to see how these crystals form and what the scallop is doing to shape them, we had to move to juvenile scallops where the crystals are not fully developed yet. At this stage, the eyes are very small. You can see them by optical microscopy, and by using cryogenic scanning electron microscopy, you can see the architecture of the whole eye with great detail. So this pseudo-colored green region at the back, here, is where the mirror region is. A close-up view shows us that instead of the ready-formed crystals that we see in the adult, in the juvenile scallops, the mirror region is full of ellipsoidal vesicles with all kinds of various features inside them. And after viewing 20 eyes and thousands of vesicles, we can put together a sequence of what's going on. To put it simply, a scallop uses a template to shape the crystal. Initially, early crystal-forming vesicles, which do not yet contain a crystal, contain two pre-assembled fibrils, these lines here pointed to in yellow arrows. When a crystal emerges, it does so in the space between these fibrils and pulls them tightly around itself. The fibrils guide the crystal growth in a specific direction while limiting its growth in the other direction. This allows a thin plate of highly reflective guanine to appear. In reality, these fibrils are sheets which chemically interact with the forming crystal and direct its path and affect its final shape. So now, armed with this knowledge, we can go back to the lab and try again. I would say it's a bit better, right? You can already see the highly reflective phase being expressed, and the crystals look far more uniform. However, there is still a ways to go. The process we used is toxic, it's not natural like the scallop, and we still need to learn a lot about the chemistry of the process, like what is the chemical nature of the template, and what is the chemical nature of the crystallization environment. Hopefully, by harnessing biological tricks for shaping molecular crystals, we can create new materials that are bio-friendly with controlled properties. Thank you. Happy to take questions.
Hi, so thank you. It was really interesting, and I mean, this is amazing, and to see the scallops as well. So you didn't really say, but I infer from your talk that you're not a biologist, right? So my question is, what methods do you use to construct these crystals? Like in practice, how do you iterate making the crystals? And do you have competition in bioengineering, like from bioengineers?
So thank you. For making the crystals, well, what we did here was we used the polymer. So that's a man-based macromolecule, unlike the scallop that probably used proteins or fats or something like that. So we used the polymer, and we're working on different ways. Like what people do is try to change the solvent, or try to use all kinds of other materials to change the environment, and this way to control everything. And if we have competition, probably. Yeah.
Hi, thank you. It was great. I was wondering, because I'm a bit familiar with, like, I think it's more in the biology side, like in the, there are mechanisms in organisms that produce cells organizing in a certain periodical pattern. So is there any analogy or like similar mechanism to these constructions of crystals, like these molecular crystals and the arrays of biological cells that sometimes form in like certain body parts?
Well, I don't know, but probably there is, because we can definitely see that there's something going on. Like here, we focused on how the crystal itself forms, but the mirror region itself is also quite organized, even before everything starts. So we see that these fibers, they stretch the vesicle in a certain direction, and that actually span the whole mirror. It creates the curvature of the crystal, so it actually directs the crystal to form this curvature, and it avoids like spherical aberrations and stuff like that. So it's quite amazing. So there is some talk beforehand within the cell of how to organize the vesicles, yes.
Hi, my name is Ben-Gurion from Hebrew University, and I was raised by a chemist and forced to learn chemistry, and I promptly forgot it all when I went to college. But I would love to ask a non-chemistry question about your research subject, the scallop, and whether you have looked at guanine in any other living creatures.
Yes, so actually when I started, I was on spiders, which is why I gave them a bit of credit. So we used to go out to the desert and find spiders to work with. And they are amazing. They use crystals, also they excrete them as nitrogen waste, and also they have them in cells underneath their cuticle, underneath their skin, which give them color. And also they use them in their eyes to enhance their night vision, so especially for like the night stalkers, so it's very cool. Yeah, so I've mainly worked on spiders and scallops.
Hi, thank you. I'm Oriane from Tel Aviv University. And maybe I didn't understand all the way from your talk, but are you trying to also kind of mimic the process that the scallop is doing in their cells? And I guess how far can you take it? Like, would it be better to try to grow the cells, but like outside the scallop, or maybe just start growing scallops for their crystals? I don't know, but thank you. It's super interesting.
Yes, thank you. So it's hard to tell how practical we can be about it because we still don't know a lot. So we don't know about the chemistry of what's going on in the scallop. And this is fascinating because guanine is a highly insoluble molecule in aqueous environments, which makes all our experiments really difficult. And we always have to resort to organic solvents, which are not very nice for the environment. So we're still trying to find out how the scallop is doing this. And once we do that, then I hope that we can mimic it. Yeah, I really hope that we could. Yes. And regarding growing cells out of crystal-forming cells, so it was tried a few years ago. To grow from fish, and the cells stayed alive for a few months, but they didn't grow, they didn't proliferate. So I'm not sure if it's possible, but there's also quite new in the field that have found a lot of microalgae, unicellular microalgae, that also can make these crystals. And then maybe that could be a way for biotechnology to catch up. Right.
Any additional questions? Thank you very much, Avital.